This is going to be the first part of chapter 25, which is about carbohydrates. The reason I've chosen to do carbohydrates next is because most of the chemistry of carbohydrates is either the chemistry of alcohols, which was chapter 17, or the chapter of 19, chemistry of aldehydes and ketones. And because it's fresh on your mind, we're going to go right on into carbohydrates now. And we're going to start with the carbohydrate structures and the nomenclature and the terminology in this portion of the lecture. And the most obvious question is, where did this term come from? And carbon and hydrogen, hydrate is kind of a term that people use for water. And so this is a combination of the words for carbon and hydrated. Carbon was what people thought maybe this formula that they found for glucose, which is C6H12O6, could be written as 6 hydrates of carbon, and it was kind of fortuitous, perhaps, or coincidental. And they thought that maybe it was a hexahydrate of carbon, and of course we know that it's nothing like that at all, and people knew that pretty soon after they figured out this more about these molecules, but nevertheless this terminology has persisted for this whole group of compounds. And we call them some other things too besides carbohydrates. We call them sugar molecules or saccharides. You'll hear all of that terminology. And the reason that these are such common molecules is they make up about 50% of the Earth's biomass, and these are the molecules that plants use for structural components as well as for storing energy or food molecules. And of course when animals eat plants, they use the structural molecules for fiber, and some of it has no nutritional value at all for animals, but the energy molecules in the plants are also energy molecules for animals. So these are very important molecules, and they are the first biomolecules that we've talked about. And there are a number of ways to classify carbohydrates, and the most straightforward one is to classify them by the number of sugar molecules. And remember, I told you that we might also call them sugar or saccharide molecules. And so if you have a simple sugar, then that means it can't be broken into smaller units that we would still consider to be a carbohydrate. And so those things might be called simple sugars, or they might be called monosaccharides. And if they contain more than one sugar molecule, then they can be complex, and they can have prefixes like disaccharides would be 2 sugar molecules, trisaccharides would be 3, and so on. And if you have a whole bunch of them in a row, then those are polysaccharides. An example would be cellulose is a polysaccharide. And another way we can organize the terminology is by calling these out based on what the carbonyl form is. All of these have a bunch of alcohols on them, but they usually only have one carbonyl, and that carbonyl could either be an aldehyde, in which case we could call it an aldose, and if that carbonyl is a ketone, we can call it a ketose. And you'll notice these OSE endings are significant to tell us that overall the molecule is a carbohydrate. We might also use a classification based on how many carbons are in the backbone. So if we have 3 carbons in the chain, which is the smallest that you could have, it would be a triose, and 4 carbons would be a tetrose, and so on. And you can also have combinations of these terminologies. So if you had an aldopentose, that would mean it's a 5-carbon carbohydrate whose carbonyl is in the aldehyde form. And if you had a ketohexose, that would be a 6-carbon chain, and the carbonyl is in the form of a ketone. And so again, all of these are going to end in the OSE ending, so we know that it's a carbohydrate. Back in chapter 5, we learned about Fisher projections, and Fisher projections are drawing the big plus sign to indicate a stereocenter. And in that convention, the horizontal lines are coming out of the plane or toward the viewer, and the vertical lines are going back or away from the viewer, and only the central atom at the at the junction of the big plus sign is actually in the plane of the paper. And when you use Fisher projections, you're allowed to do two types of rotation that don't change the sense of the molecule. One is that you can rotate it 180 degrees in the plane of the paper, and I told you, you could just turn the paper 180 degrees. And the other is you can hold one of the groups and rotate the other three. And we did a number of exercises with that kind of thing. This is one point where if you have models, you should get them out to just review this, because it will help a lot. So once you have a Fisher projection, what you need in order to determine whether your stereocenter is R or S is you need for the lowest priority group to be at the top. And that's why you need these allowed rotations, because sometimes they're not drawn that way initially. And so you have to do an allowed rotation to get the lowest priority group to the top. And then after you have the lowest priority group to the top, remember that it's on the vertical line, it's behind the plane of the paper. And so you imagine that you're holding onto that group, and then you let your eye rotate around from priority groups 1 to 2 to 3 and decide whether your eye would be traveling around those three groups clockwise or counterclockwise. And if your eyes are traveling clockwise around that, then it would be R, and if it's counterclockwise, it would be S. So we'll start with the simplest sugar molecule, which is glyceraldehyde, and every carbohydrate contains at least one stereocenter or chiral center, and glyceraldehyde only has one. And in the conventional way of drawing these things, the biochemists like to draw them where you put the carbonyl group at the top, and the thing goes vertically from there. And of course, in the case of glyceraldehyde, there's only 3 carbons, so we're going to let the carbons go up and down from there. So the aldehyde group is at the top here, and the other end of the chain is at the bottom, and so I have this 3-carbon chain. And so this is a conventional way of drawing it. Well, if I want to determine whether this carbon is R or S, I need to do an allowed rotation. I need to get this lowest priority group to the top. So if I hold group number 3 and leave it at the same place, then I'm going to move 4 to the top, and the aldehyde group's going to come over here, and the OH is going to go over there. So that's what I mean by hold 1 and rotate the other 3. Once you've done that, then you can decide, is it RS. So now your lowest priority is at the top. You kind of ignore that, and your eye goes from 1 to 2 to 3, going around that. You see that's clockwise, so this one is R. And so we know that original drawing over here, this was R. And so you can see that included in the nomenclature. Now, the fact that it's drawn in the R configuration doesn't tell us what direction the light will be rotated. Remember, stereocenters were discovered because they cause the rotation of plane polarized light. And if you have a mixture of enantiomers, that they rotate light in exactly the same direction but in opposite directions. And so if you had a mixture, an exact 50-50 mixture, that would be called a racemic mixture. That mixture would not rotate the light. But if we can separate the enantiomers, then we can see the rotation of the light. And if it rotates, if the molecule rotates the light to the right, it's called dextrorotatory, or we use a D or a plus sign for that. And if it rotates it to the left, then it's levorotatory, or we use an L or a minus sign for that. And these things are not intrinsically related to the nomenclature. The direction of the rotation of the light and the nomenclature are really derived from completely different methods. And so, as it happens, because the guy who figured this out was Emil Fischer, he was pretty smart, he kind of got it right. And so, as it happens, that R-glyceraldehyde rotates light to the right. And so his convention of how he figured out, how to draw it, turned out to be coincidentally, or maybe it was intuitively correct, that the R molecule does rotate light to the right. And so we could also call it D-glyceraldehyde because it's rotating the light to the right, or use the plus sign for it. And since the nomenclature R doesn't intrinsically tell us which direction the light is, but if we wanted a really complete name that tells us both about how to draw it as well as how it rotates the light, we can put the R and the plus sign out in front of the name, and that would tell us both pieces of information. As it turns out, all of the common sugars are D sugars, that is, they all rotate light to the right. And as it turns out, they're all going to have the R configuration at one particular place in the molecule. And so we'll see that when we look at another sugar molecule. So here's glucose, which is common name is also dextrose. And so I've numbered the carbons here and drawn it in the conventional way that biochemists like, which remember was to put the carbonyl at the top and let the chain go vertically down. And so when we number the chain, remember aldehydes take priority, so this is going to be carbon 1, and there's 6 carbons, so down to 6. And if we do the priorities on this carbon right here, which is kind of in the same position as the one that we did for glyceraldehyde, then you see when you do the priority numbers, and if you do your allowed rotation, you're going to find out that this carbon is also in the R configuration. And we call this carbon right here the penultimate carbon. It's always the next to the last carbon. And for all of our common sugars, this carbon is always going to be R. And as it turns out, all of these are stereocenters, and all of them contribute to the optical rotation, but almost all of the sugars still end up rotating light to the right. Even when you summarize all the effects of all these stereocenters, they still end up rotating light to the right. Okay, so let's review a little bit. If we go back to chapter 19, which we've just recently done, and look at an aldehyde, so here's a generic aldehyde, we talked about acetal formation, and that started out with protonation, and then there was the resonance structure, which would put positive charge on the carbon, and then an alcohol molecule could react with that carbonyl, and when you put a third bond on the oxygen, it gets a plus charge. If we lose that proton off here, we get to this species right here, which is called a hemiacetal. And I told you that in a biological system, it would be common for them to stop there. In a lab, we don't usually stop at the hemiacetal, but in a natural molecule, in a biological setting, it's very common for them to stop at hemiacetals. Okay, so what that has to do with carbohydrates is that a lot of carbohydrates exist in a cyclized form, which is actually a hemiacetal, because in this R group there are alcohols down the chain, and so they self-react, and it's called an intramolecular cyclization. Now, these two molecules are drawn on here because the terminology is going to reflect these terms. So this molecule is called pyran. It's not aromatic, but we did talk about it in what it would take for it to be aromatic, but that turns out not to really have much to do with carbohydrates. And we also talked about furan, which is aromatic, and again, that doesn't really have anything to do with this particular chapter, but that's where you've seen these molecules before. So we're going to talk about sugar molecules that make a 6-membered ring, and the 6-membered ring that it makes is going to be called a pyranose ring because it's similar to this without the double bonds. And if they make 5-membered rings, they're going to be similar to this. They're going to be called furanose rings, but again, not any double bonds. And there's an equilibrium just like this between the open chain form and the cyclized form. So going back to this carbon, remember this carbon number 5 was the penultimate carbon, and so here's another reason why it's important. Not only does it have the R configuration, it also uses this oxygen to close up and make the cyclized ring. So the intramolecular hemiacetal formation, you see the numbers are on this structure also. This carbon right here, carbon number 1, is still carbon number 1 over here, and when you make the hemiacetal, that carbon has both 2 oxygens on it, and in the hemiacetal, one of them is an OH and the other one is part of an ether. All right, and so when you number these carbons around here, then this This hemiacetal is the cyclic form, and it's called a pyranose ring, and it came from glucose, so it's a glucopyranose ring. It specifically came from D-glucose, which is what I have drawn here. And then we have this other terminology, beta. Now look at your two structures here. Here's another one. So if this guy comes around and closes from the top, or it could come around and close from the bottom. Remember that I told you that carbonyls are planar, and so the nucleophile can approach from either the top or from the bottom. And so if it approaches from one side, you're going to get what's called the beta, and that has to do with the fact that this OH is pointing up as opposed to pointing down. And so this cyclized form is where the OH is pointing down is called the alpha D-glucopyranose, and we've got this thing drawn with the OHs in equatorial position. This one is axial, whereas all of them are equatorial when it's in the beta form. It probably won't surprise you to realize that the beta D-glucopyranose ring is a little bit more stable because all of the OHs are equatorial, and alpha is not quite as stable because of the steric crowding problem. So this one is a little more favored than that one. And also, don't lose sight of the fact that this carbon number 1 is really what was the carbonyl in the open chain form. And in a biological system, these things are in equilibrium and going back and forth all the time. So here's another example. So this one is called D-mannose, and if you look, the difference is in the position of this one OH. So let's go back. So in glucose, you've got 3 OHs on the right-hand side and 1 on the left. In mannose, you have 2 on the right and 2 on the left. But you notice the penultimate carbon, the molecule is still in the R configuration on that particular carbon. And something else you'll see in this drawing, they've used a little bracket here, and they've put hydrogen comma OH, and what they mean is they're showing you a shorthand form where there's both forms. The alpha, remember the alpha one would be here for the OH, and the beta one where the OH would be up here. Both of those are formed, and this equilibrium's going back and forth. Something else, when you go from the open chain form to the cyclized form, the sort of the duh thing for how you draw this is that out of these things that are on the left, they will be up in the cyclic form. So 2 of these guys are on the left, and so remember the numbering system, this is 1, 2, so on number 2 and 3 the OHs are up. And so on number 2, the up position is axial, and on number 3, the up position is equatorial. And so that's how you know. And if we go back again to this guy, all right, remember only one of them was on the left here, and that's on number 3, and that one's up, and up is equatorial in either form of this ring. You should be able to do that. You should be able to go from an open chain form to a cyclized form, and remember, left goes up, and you have to keep straight whether it's axial or equatorial, because remember those things alternate. So fructose is the 6 carbon molecule that is a ketose. So carbon number 2 is a ketone, and it turns out it can make either 5 or 6 membered rings. Actually, the 5-membered ring is one that you'll see a lot with fructose, even though it's not necessarily the most common. And in this case, if it uses the penultimate carbon, then it's going to make the 5-membered ring, and so they've drawn that one here. And that one looks a little different than the 6-membered ring, and so it had to use the other oxygen at the end of the chain to make the 6-membered ring. And you still have a carbon number 2 was the carbonyl, so it's still a hemiacetal. And carbon number 2 here, you see it's still a hemiacetal. It's got an OH and then an ether-like position on it. And carbon number 3, there's an OH on the left, and so when you look at that, there it is, it's up here, and it's up here, so that part is still true. And here's the picture of furan, which was the parent name for the furanose ring, and we've already seen the pyranose rings here. So either one of those is a possibility. Something else I want to point out too, when we've got both of these on the same slide, when biochemists are drawing these cyclic forms, they always put the oxygen in the ring in this position or in this position if they're talking about a furanose ring. Now, the fact that it makes these rings in these hemiacetals is disguising the behavior of the carbonyl. We talked about using acetal formation in aldehyde and ketones chapter to talk about disguising or protecting the carbonyl. And we also talked about the fact that these hemiacetals could react again and go to a full acetal. And when we make these cyclized forms of these sugar molecules, we've created another stereocenter. And what that does to the optical rotation is it kind of messes it up. Because remember, it could make either the alpha or the beta stereocenter there at that point. In other words, the nucleophile, which is the oxygen, could come from the top or the bottom. And because you've got a mixture there, it blurs the optical activity. The other ones are still the same, but it still makes it kind of fuzzy at that point. And so that blurring of the optical rotation is called mutarotation. And it's a rapid equilibrium, so it's kind of opening and closing. And so instead of looking at it in solution and seeing a sharp line for whatever its optical activity is, you see a blurring. And so that is called mutarotation, and that carbonyl carbon, where that's going on, is called the anomeric carbon, or the anomeric center. And the two isomers, remember, were the alpha and the beta for those two diastereomers, and so they're also called anomers. So the molecules that I want you to know, both the names and the structures for, in either the open or the cyclic form, are pictured in your book on page 1009 in the 8th edition book. Glyceraldehyde we've talked about, glucose we've seen, and mannose we've seen. You should also know D-ribose, which is a 5-carbon sugar. Fairly obviously that's important in DNA and RNA. And galactose, and you'll see why that's important a little later. And fructose we've also talked about on page 1013. And there's a couple other places in the book where they're pictured. So for this section of the chapter, you should know both the names and the structures, both open and cyclic, of the molecules that I've mentioned on the previous slide, and you should be able to either draw them or to recognize them. You should know all the terminology that we've talked about so far with mutarotation and anomers and various kinds of oses and such, disaccharides and all of that terminology. And you should be able to figure out R or S configuration if you have a Fisher projection and be able to recognize stereocenters. And that is the end of this section of the book.